Hello again. We're going to continue talking about metabolism, specifically now amino acid metabolism. So we had a little complication uh, to this now because amino acids contain this nitrogen group that we have to do something with. So that's going to be a main complication. But I just want to show again this diagram out of the textbook and of many of the reactions involved in metabolizing different types of compounds. And the shaded areas in this diagram indicate where reactions involving nitrogen containing molecules are. So you can see that takes up a large portion of this diagram, so a lot of reactions. And that's because, as you know, there are 20 different amino acids, and they all need to be metabolized just a little differently. So what we're going to do in this session is talk about a little bit about amino acid synthesis, degradation. We'll also talk about derivatives of amino acids, some of the other compounds, uh, vital compounds that are derived from amino acids, and then how to get rid of excess nitrogen via the urea cycle. Before we get into amino acids specifically, I just want to review some of the overall ideas about energy metabolism. And when we talk about energy metabolism, we're talking about the sum of all chemical reactions that go on in the living cell. And the generation of energy obviously is needed for a number of different reasons to maintain body temperature, uh, in muscle movement, uh, sending electrical signals to control all this, as well as in breaking down other chemicals and synthesizing important chemicals in the body. So that all requires energy. So just a couple of definitions to remind you when we talk about anabolism, we're talking about the building of molecules. Generally that, of course, is going to require energy. The breakdown of molecules, catabolism, releases that energy. And remember, when we're releasing energy, we're going from a more reduced state to a more oxidized state. And the final product of that oxidation, fully oxidized carbon, is carbon dioxide. Fully reduced state would be a simple hydrocarbon like methane or ethane. Okay, so we can break down or build up the three major types of molecules in our bodies: carbohydrates, fats, or proteins. And again, in order to build up those, we do require energy. Breaking them down then yields energy, whether we're breaking down sugars or fats or amino acids that came from proteins. They all will yield um, energy, ultimately in the form of ATP. Now, a lot of this takes place in the liver. When if you're not sure where things are happening, a good guess is always in the liver, um, and that's where a lot of different reactions take place for all different types of molecules. As far as carbohydrates, although you can make carbohydrates or break them down in other tissues, really the liver is responsible for converting fructose into glucose. So if we eat a lot of fructose in our diets, that it has to be digested pretty much in the liver. And we'll get back to that later on in the semester and find out when we talk about what can happen if you have a little too much fructose in your diet. Liver is also a place where we can store some carbohydrate, not a whole lot, but some in the form of glycogen. And so, of course, it's going to be responsible for breaking down glycogen and releasing glucose molecules that then can be utilized for energy. Also, that's where the majority of glucose is synthesized through gluconeogenesis. And we can also convert glucose into fatty acids through glycolysis, converting it into acetyl-CoA, and we can make fatty acids from that. And remember, and I will be mentioning this again and again, that we cannot go from fatty acids to glucose. Once we get to the 2-carbon stage, cannot go back up to gluconeogenesis. Now, the liver is also responsible for breaking down fats, whether it's fatty acids or more complex lipids. You can package extra lipids for transport to other body organs. It's where bile is manufactured and sent to the gallbladder. And also, we can make ketone bodies out of lipids in the liver. Now, proteins also are metabolized to a large extent in the liver, and we can manufacture the non-essential amino acids. Those are the ones that we are able to manufacture. Can remove those amino acids that are present in excess. So we have 20 different amino acids, and they are utilized to different extents in terms of incorporating them into proteins, but we do have to maintain a balance of amino acids. So if we have an excess of one amino acid, it can be digested, and that mainly occurs in the liver as well. Removal of ammonia from the bloodstream and converting it into urea, which is then sent to the kidneys, also occurs in the liver, as well as making other nitrogen-containing compounds, the ones listed here, RNA and DNA. It also carries out a number of other functions, detoxifying alcohol and other drugs and other compounds. It helps in getting rid of old red blood cells. We can store a small amount of some vitamins and minerals, and also activates vitamin D. So a lot going on in the liver, including the metabolism of amino acids. Okay, so these are the backbones of the 3 major sources of energy: glucose, or many other sugars. It has a 6-carbon backbone. Fatty acids have a much longer chain and are much more reduced. And amino acids, which can have various length side chains, and they all contain this nitrogen in the form of an amino group, which has to be removed before it can be digested to produce energy. Okay, so since all the amino acids are different, have different R groups, as you might imagine, they're going to feed into different pathways. Some amino acids feed into pyruvate. When that happens, the pyruvate can either be used to synthesize new sugar via gluconeogenesis, or it can be sent through the TCA cycle by converting it to acetyl-CoA. Other amino acids are converted directly into acetyl-CoA and feed into the TCA cycle. And again, if that's the case, we cannot go backwards and make glucose. So we cannot make glucose out of those amino acids that feed directly into acetyl-CoA. Other amino acids feed into intermediates of the TCA cycle. And again, we wouldn't be able to synthesize glucose from those. So this diagram pretty much shows the same thing. We see that in the middle, the blue boxes are metabolism of carbohydrates, first into glucose or other monosaccharides, and then eventually into pyruvate through glycolysis, and then acetyl-CoA. Now, we see in this diagram the arrows are only pointing in one direction, but of course pyruvate can be reversed and made into glucose, and that then can be made into carbohydrates. It's the last step, going from pyruvate to acetyl-CoA, that is not reversible. If we look on the left, we see the proteins first broken down into amino acids, some of which feed into pyruvate and can be converted into glucose. Others feed directly into acetyl-CoA or straight into the TCA cycle through intermediates of the cycle. If we're breaking down fat, the fatty acids feed directly into acetyl-CoA, can produce a lot of energy with those, but we can't go backwards and make carbohydrates out of them. Glycerol feeds into pyruvate, so we can convert glycerol into sugar. However, that's a pretty minor component of fat. Okay, once this feeds into the TCA cycle, then eventually we're going to go through the electron transport chain and produce ATP. Okay, so again, some amino acids and glycerol can be converted into pyruvate and therefore glucose, and that's needed. We have to have a certain level of circulating glucose in the bloodstream in order to maintain healthy central nervous system and red blood cells. So without glucose, the body's going to start to break down tissues, particularly proteins. So the intake of adequate carbohydrate will prevent this. And again, fatty acids are converted directly to acetyl-CoA, cannot be used to make glucose. So once we break down glucose into pyruvate, again, a number of things can happen. It can be used for quick energy by converting it into lactate. And that does not require oxygen. So that's an anaerobic process. But we cannot maintain that for very long because there just isn't a big supply of carbohydrate. So eventually we have to go to sending the pyruvate through the TCA cycle via acetyl-CoA. And that again is an aerobic process requiring energy. This is a much slower process than glycolysis, so we produce much more energy that way. But if we need quick energy, the way to get it is through glycolysis. And the last time we talked about or maybe 2 times ago, we talked about the Cori cycle. If tissues like muscle is breaking down pyruvate very quickly, converting it to lactic acid. That lactic acid then can travel through the bloodstream back to the liver, where it can be converted back into glucose via gluconeogenesis. Remember, this uses up a good deal of energy in order to do that. And this is a diagram, again, of breakdown of glucose in the muscle tissue to produce pyruvate, which then has to be converted to lactate to generate cofactors for glycolysis. And then that lactate can travel to liver, be converted back into glucose, which then can be sent back to the muscle for breakdown again to generate more energy. So um, this is another example of a similar cycle. Again, we may call this Cori cycle. Um, and that, in this case, using alanine to transport the energy from the muscle, or to transport molecule from the muscle to the liver, where it can be converted into glucose, and then that can be transported back to the muscle. So if the cell needs energy and there's plenty of oxygen available, pyruvate can enter the mitochondria, where it can be converted to acetyl-CoA and then sent through the TCA cycle. Again, this is an aerobic reaction requiring oxygen. If there's a limited amount of oxygen, that pyruvate then can be converted to lactate and sent back to the liver to be resynthesized into glucose again. And remember, some bacteria, particularly oral bacteria, prefer to just stop the process at lactate, secrete the lactate out into its environment, and break down more sugar. And of course, this produces a very low pH in the local environment, which can start to erode the enamel on your teeth. Okay, so a lot of this centers around pyruvate. Again, you can see that from this diagram, pyruvate's in the middle here. You can use it to make glucose. You can break down glucose to make pyruvate. So that's a two-way pathway, two-directional pathway. You can then convert the pyruvate to acetyl-CoA. That's a one-directional reaction, so you can't go backwards there. We can also feed some amino acids, which we term the glucogenic amino acids, into pyruvate because they can ultimately be used to make more glucose. Other amino acids, the ketogenic amino acids, feed directly into acetyl-CoA. Now, once we get to acetyl-CoA, it can either go into the TCA cycle, not shown here, or it can be used to synthesize fatty acids. And again, that's a two-directional pathway, so we can make fatty acids or break them back down into acetyl-CoA. Also, we can convert that pyruvate into lactate to generate more NAD. And that then lactate can be converted back into pyruvate, generally if it travels back to the liver. And again, if we're breaking down lipids, triglycerides, the glycerol backbone can be converted into pyruvate, whereas the fatty acids, of course, cannot. Okay, this is a table showing how much energy is available from these different types of molecules, and I don't know where these numbers come from. I think when we look at the top three molecules here, actually it looks like two molecules, is glycogen in the liver and muscle, and free glucose, and those are pretty constant from person to person. But the amount of triglyceride and the amount of protein can vary quite a bit from one person to another. And again, we have a limited capacity to store carbohydrates in the form of glycogens, some in the liver and some in the muscle. But when you look at the number of calories you can generate from that stored amount of carbohydrate, it's pretty small. We also have a small amount of glucose circulating in the blood that has to be maintained, but there's not a whole lot there either. We can store a, a huge amount of lipids in the adipose tissue. And like I said, I'm not sure where this number came from, if this is what's considered a typical or an average or what. Um, uh, but the other thing is we really don't have a place to store protein or amino acids. It gets incorporated into muscle. We can break down muscle if we need those amino acids, or if there's no carbohydrate and we need to break down amino acids for energy, we can start to break down muscle, but it's not really a storage compartment. So there is no dedicated storage for protein or amino acids. Now this says most amino acids. I would say many amino acids can be converted into glucose, but before that can take place, they have to be deaminated. In other words, the amino group has to be removed. So we can use those amino acids then to make pyruvate, then can be used to synthesize glucose, which can be used to maintain a central nervous system, red blood cells, or wherever else it's needed. And again, some amino acids are converted into acetyl-CoA. Those can either be converted into fatty acids or enter into the TCA cycle. Okay, so this diagram shows the same thing. We have some amino acids shown in the top of the red box that are converted into pyruvate, and then others shown below that are either converted into acetyl-CoA or into TCA cycle intermediates. Okay, so dietary protein is the main source of amino acids, and we're only going to start breaking down muscle if we don't have dietary protein or other nutrients. So we can use them as fuel if we need to. And of course, we start by breaking the proteins into free amino acids, and that starts in the digestive tract, and then those are absorbed and passed into the circulation, where they go to the liver, of course, so that they can be deaminated by a transamination reaction. So then we're left with these alpha keto acids that can be used as a fuel or as an intermediate to make other compounds. So again, unlike carbohydrates, which have a small amount of capacity for storage, and lipids, which have an unlimited amount of storage capacity, amino acids do not have a dedicated storage form. And then, of course, if we're breaking down amino acids to produce energy, we're going to produce an excess of nitrogen, which has to be ultimately excreted. Okay, so this shows the pathway of digestion of protein, again, starting in the mouth, going in the stomach, where it starts to be broken down into smaller peptides, goes through the intestines, where it's broken down finally into amino acids, sometimes dipeptides, and then those can be transported in the bloodstream to the liver, where they can be utilized either to make new proteins or as energy by breaking them down, feeding them into either glycolysis or the TCA cycle, or they can be used to synthesize other nitrogen containing compounds. Now, when we talk about amino acids, we sometimes uh, group them into two groups: the essential ones, which we aren't able to synthesize, so we have to get them from our diet, and the non-essential ones, which we are able to synthesize ourselves. Um, Keep in mind that this is a fairly fluid definition, because if you were to look at another table of these, you might find some slight changes. I mean, some of them, like glycine, alanine, serine, are always going to be in the non-essential groups, but others are a little ambiguous. For instance, we have in this table at the bottom of the right tyrosine, um, which is in the non-essential list, which means we can synthesize that. But we synthesize that from phenylalanine, which is over on the other side in the essential group. So we can't make phenylalanine. So if we don't have phenylalanine, we're not going to make tyrosine. And this table, arginine is in the essential group, but we can make arginine through the urea cycle from other compounds. So again, that's the one that's a little bit ambiguous. Okay, this diagram shows the many different pathways for amino acids, starting up in the top left where we see an ingested protein, which is converted into amino acids. It can go to the left here to make proteins that we need, or we can break down some of those proteins into amino acids if we need them. Then those amino acids, if they're not used to synthesize new proteins, can be used to make other nitrogen-containing compounds. There's a list of some of them here. And again, they can be funneled into either the TCA cycle or glycolysis to generate energy. But they ultimately then, we need to get rid of the amino group first by removing it as free in a two-step reaction, actually, as we'll talk about in a minute, as ammonia, and then converting that into urea. Okay, again, each of the 20 amino acids needs to be degraded in its own way because they're all different. However, they all have a certain amount in common, and that particularly is the removal of the amino group by a transamination reaction. Okay, so again, we don't store amino or nitrogen containing compounds. So if we're breaking down protein and amino acids, the amino nitrogen has two possible fates. It can be used in the synthesis of other nitrogen containing molecules, and there's a list here. What's not on the list is other non-essential amino acids. We can use the nitrogen from other amino acids to, if we have a shortage of certain non-essential amino acids, or if the amino acid is going to be broken down to produce energy, the nitrogen must be incorporated into urea and excreted. So again, the first step in breaking down an amino acid is the removal of the amino group. This is called transamination, and it involves a transfer of the amino group of any amino acid to the keto group, generally of an alpha keto acid. There are a number of transaminase enzymes. Most of them use alpha ketoglutarate as the receptor of the amino group. Acceptor, I'm sorry. and that converts the alpha-ketoglutarate into glutamate, which of course is one of the 20 amino acids. So you might be asking, well, we really didn't get anywhere here. But as you'll see, we can break down glutamate to release ammonia. But let's look at a couple transaminase reactions first, just to make sure we understand what's going on here. And in this example, we start with the amino acid aspartate, which is an acidic amino acid, has an acidic R group. And we then are going to transfer the amino group to alpha-ketoglutarate to produce oxaloacetate, an intermediate in the TCA cycle, and glutamate. So we've broken down, we've converted aspartate into oxaloacetate with the production of more glutamate. So eventually we'll have to do something about the glutamate. Another example is the amino acid alanine, which you might remember the R group is a simple methyl group. I'm not sure why in this diagram they called alpha-ketoglutarate by another name, acceptable name, but not one it's usually called, oxoglutarate. But again, the amino group is transferred to the keto group, and in this case, producing pyruvate and again glutamic acid. So now pyruvate then can be utilized to synthesize new glucose. So we can feed alanine directly in to gluconeogenesis by converting it directly into pyruvate. So these transaminase reactions are reversible. They can go easily in either direction, and depending on the concentration of the different amino acids, they may go one direction or the other. And so if you have a lot of one amino acid, even though it may not form a direct transamination with another one, you can transfer that amino group to alpha-ketoglutarate, producing glutamic acid, which then can transfer the amino group back to a different amino acid. So in the process of oxidative deamination, the glutamate amino acid group is removed as ammonium ion, and that converts the molecule back into alpha-ketoglutarate. So the transaminases can equilibrate these amino groups among available alpha-keto acids to make different amounts of amino acids. So we can modulate these different non-essential amino acids to maintain the amount that's needed in order to synthesize new proteins. So even though we can convert one amino acid into another, we can use the nitrogen on one amino acid to generate another different amino acid. Now that nitrogen has to be obtained in the diet as amino acids. So this is the general scheme then of breaking down amino acids, converting them into alpha-keto acid, then depending on which amino acid it is, feeding it generally into the TCA cycle or glycolysis, pyruvate. That amino group is generally transferred to alpha-ketoglutarate to make glutamate. So in the diagram, the orange line follows the amino group. Then that glutamate can be converted back into alpha-ketoglutarate by releasing the ammonia, which then feeds into the urea cycle, and is converted into urea. So ammonia is highly toxic to living organisms, so we have to eliminate it pretty quickly. We can't let it build up. And different types of animals will excrete ammonia through different mechanisms. Fish can excrete it directly as ammonia through the gills. Some animals, particularly birds, convert it into uric acid, which is a nucleotide derivative, a purine derivative, which we'll see later. But we convert our ammonia into urea for excretion. And just as an aside, those of you who have a swimming pool, if you ever have a algae bloom, one way to get rid of that is by adding high concentrations of ammonia along with chlorine in the form of bleach. And chlorine facilitates the uptake of ammonia by the algae, which then kills it. It's a nice way to get rid of it because it's a non-toxic compound. It is volatile. It burns off very quickly, unlike some of the fungicides that people use in their pools. Anyway, back to amino acid metabolism. So again, this all takes place in the liver, and after urea is formed, it's transported to the kidney, where it can be transferred to the urine for excretion. So this is the urea cycle. We're going to come back to this. I'm not going to look at this in detail at the moment, but we'll come back in a few minutes. But basically, we're converting the free ammonia, combining that with a molecule of carbon dioxide and phosphate from ATP into this carbamoyl phosphate that then is added to ornithine. And then we eventually, from that, make arginine, which we can then break off the urea, which then can be excreted, forming ornithine then, which can accept another molecule of carbamoyl phosphate. Okay, so any amino acid destined for energy metabolism has to be deaminated to yield the carbon skeleton. And again, it's done by a transamination reaction, which transfers the amino group from the amino acid to an alpha keto acid, generally alpha ketoglutarate, to make glutamate. Then that can be deaminated directly to give free ammonia. So this is the reaction for oxidative deamination. So in addition to removing the amino group from the glutamic acid to make alpha ketoglutarate, we do generate a molecule of NADH from NAD, which is a high energy molecule that can go into the electron transport system to produce ATP. So this is an energy producing reaction. And in addition to that, of course, we are left with free ammonia. So this is showing that reaction again. Remember two things about this reaction. One is the reduction of NAD or NADP to make the higher energy NADH, and the generation of free ammonia. We can also generate pyruvate directly from serine with the release of free ammonia. And that's through using the enzyme serine dehydratase. So serine can also be used to generate this free ammonia that can then feed into the urea cycle. Okay, so in summary, in order to break down amino acids, we need to transfer that amino group from the amino acid to alpha keto acid. Generally, that's alpha ketoglutarate, which is converted to glutamate, and then glutamate can, through the glutamate dehydrogenase, lose that amino group, converting it back into alpha ketoglutarate and generating a little bit of energy through the form of NADH at the same time. Okay, we can measure the amount of blood urea through laboratory test called BUN, or blood urea nitrogen. You can monitor patients with amino acid metabolism disorders or renal problems using this method. And there's a colorimetric method for this as well. Okay, we've been talking about converting these amino acids into, or transferring the amino group to alpha ketoglutarate to make glutamate. And glutamate is a very common molecule sold in grocery stores and often used as a food additive in Oriental and other restaurants. An excess of monosodium glutamate, or MSG, can cause some temporary symptoms: weakness, tingling, sensation of warmth in the face and upper torso. But this generally doesn't last. It's pretty transient. And some people are more susceptible to this than others. There's generally no permanent damage from MSG, although it can trigger bronchospasms with people with severe asthma. And the reason for this is that glutamic acid is also a neurotransmitter, and it's an excitatory neurotransmitter. There is a similar molecule that's a breakdown product of glutamic acid, gamma aminobutyric acid, just missing the carboxyl group. That's an inhibitory neurotransmitter, so it kind of has the opposite effect. So I want to talk briefly about three molecules that have the same carbon backbone shown in the top line: alpha ketoglutarate, glutamate, and glutamine. Alpha ketoglutarate again has no nitrogen or amino group, glutamate having one, and glutamine, another amino acid, having two amino groups with the amide R group. Again, the enzyme glutamate dehydrogenase transfers the amino group from glutamate to removes the amino group from glutamate to produce alpha ketoglutarate. And remember, that's a reversible reaction, so it can go in either direction. Glutamine can also be formed from glutamate by adding ammonia. This is an energy requiring reaction. And glutamine can be used as a sink for excess ammonia, and then it can be converted back into glutamate using a second enzyme, glutaminase. As you can see from this slide, nitrogen enters the urea cycle from all the different amino acids, starting up in the upper left-hand corner here. Again, those can all transfer the amino group to alpha ketoglutarate, making glutamic acid. Can also generate aspartate through aminotransferases that again feeds into the urea cycle to add another ammonia. So two ammonias are added to the cycle each time to make the urea, which again has two nitrogens and one carbon. One coming from free ammonia that's been cleaved off of glutamic acid to give us alpha-ketoglutarate back. That then is made into carbonyl phosphate, which combines with citrulline or with ornithine to make citrulline. And then we add the second amino group from aspartate, which forms a covalent bond with the citrulline, and then it's released, leaving the amino group there. At that point, then arginine is formed, and then that can be cleaved to produce urea, and we get ornithine back. Again, there are two different reactions to deaminate amino acids. One is the oxidative deamination, which we've talked about at length, transferring the amino group of one amino acid to alpha-ketoglutarate, generating glutamic acid. The other is a non-oxidative deamination, which is using two amino acid substrates, serine and threonine, both of those which contain hydroxyl groups, and generating, again, free ammonia. So it's important to balance the amount of ammonia in cells, and this shows a number of sources on the left, and you can read through those, although we've talked about most of those, and then utilization on the right, and basically to form different amino acids, form other nitrogen-containing compounds, and then if there's excess ammonia left over, that is made into urea that then can be excreted through the kidneys. Again, if we look at the urea cycle in this figure, this is kind of a bare bones figure, and just again want to point out that this starts in the mitochondria where carbon dioxide combines with free ammonia and a phosphate off of ATP, although this reaction requires two ATPs to make carbonyl phosphate. That then combines with ornithine to make citrulline, which then goes out to the cytosol for the rest of the cycle. And again, we're going to revisit carbonyl phosphate synthetase, which is found in the cytosol. It's a completely different enzyme. It carries out the same reaction, but that reaction is used in a different pathway in nucleotide metabolism. So keep in mind the connection between the urea cycle and the TCA cycle, and that is mainly through aspartate losing an amino group to become fumarate, and again, fumarate being an intermediate in the TCA cycle. So we can feed amino acids directly into the TCA cycle at the same time as sending the nitrogen into the urea cycle to ultimately get rid of it. Now, alanine and glutamine are responsible for the majority of amino acid movement in the bloodstream. And so they account for about 50% of amino acids released by muscle into the blood. So the amount of alanine and glutamine released far exceeds the proportion of these amino acids in muscle proteins. So there's a large amount of remodeling of protein-derived amino acids, and they occur by these transamination reactions. So remember, there's no form of storage for nitrogen-containing molecules. So if we ingest an excess amount of nitrogen, we need to remove that by first, because ammonia is very toxic at high levels. And so we need to remove that by formation of uric acid uh, and then excretion through the kidneys. So again, most mammals, including humans, get rid of nitrogen by formation of urea, but some other animals, particularly fish and some aquatic vertebrates, can get rid of free ammonia directly. Birds and reptiles make uric acid, shown here in the bottom right corner, which is a derivative of purines, which are found in DNA and RNA. Okay, I want to go through the urea cycle again, and just a few things about it. It's really the first metabolic cycle discovered in 1932 by Krebs. Again, each round of the urea cycle gets rid of two nitrogen molecules, one that comes from free ammonia forming carbonyl phosphate. That ammonia generally came from glutamic acid, and the other ammonia from aspartic acid. And then the carbon found in urea came from bicarbonate, or basically CO2. So again, this is the urea cycle, and once again, start by forming carbonyl phosphate, and that is done in the mitochondria. Again, there's another carbonyl phosphate synthetase enzyme found in the cytosol, which is involved in nucleotide metabolism, which we'll talk about in a subsequent discussion. Once that's made, it can combine with ornithine to produce citrulline. That then goes into the cytosol, where it goes through the rest of the cycle, generating arginine, which then can be cleaved to remake ornithine and as well as urea. And then that is transported to the kidneys for excretion, the urea is. And this shows the initial reaction, the formation of carbonyl phosphate. We don't need to go into any details here, but just realize that's how we initiate entry into the urea cycle. Again, I would just want you to understand that there is a direct relationship between the urea cycle and the TCA cycle, and that's shown here. I'm not going to go into any more detail since we've already talked about it. Now, the urea cycle is regulated by the formation of carbonyl phosphate, and that is done through this molecule and acetylglutamate. So there's a acetate attached to the nitrogen on glutamic acid. As far as I know, this acetylglutamate has no other function than to regulate the formation of carbonyl phosphate. So seems to be its sole purpose. However, without the presence of this molecule, the N-acetyl glutamate, we get very little or no formation of carbonyl phosphate. As you can imagine, if you disrupt the urea cycle or have a problem with the urea cycle, it can be pretty catastrophic. Ammonia is going to build up very fast. It's toxic, particularly in the brain. It can very quickly lead to a coma. You can see this if there's liver failure, because if you don't have a liver, healthy liver, you're not going to be able to generate urea. You don't see many genetic diseases that knock out the urea cycle because it is lethal. However, there are some partial deficiencies, and those can lead to some serious problems, particularly retardation. These people can be treated with low protein diets so that you don't build up an excess of ammonia. You can also treat it with keto acids to try to suck up all the ammonia and make amino acids. These are some of the defects that we see in the urea cycle. I'm not going to go into them in detail, but you can see it pretty much every step of the way. You could have a defect, and remember now, these are partial defects, so you still have some activity, because if you knock out all the activity, that would be lethal. These are a couple of drugs that are used to treat defects in the urea cycle, and the idea again is that these will react with nitrogen to form a new nitrogen-containing compound, and both of the products of these reactions are readily excreted through the urine. Arginine also serves as a precursor of nitric oxide, and the reactions are shown here, although that's really not terribly important. NO acts as a short-lived messenger for regulating blood pressure and clotting, as well as neurotransmission. One more example of amino acids, in this case arginine, as a precursor for other nitrogen-containing compounds. Now again, remember there are essential amino acids and non-essential amino acids. In other words, those that we're not able to make ourselves, so we have to ingest them, versus those that we can make, although if we're ingesting plenty of them, probably won't be making too many of them. And again, this is somewhat of a fluid idea, because, and if you look at different lists from different sources, you'll see slight variations. Obviously, there are some amino acids, alanine, for instance, that is clearly a non-essential amino acid, but depending on your definition, tyrosine could be considered an essential or a non-essential amino acid. Arginine is the same. In this figure, it says arginine is essential in infants and growing children, but not in adults. Not sure why that is, but I think that's from the textbook. So again, these amino acids, when we break them down, can feed into a number of different intermediates shown here, and this is more detailed schematic of that. Some feeding into different intermediates in the TCA cycle, and of course many amino acids feeding directly into pyruvate, and of course those then can be used to generate new glucose. Now if you look closely at this, you'll see that there's some redundancy here. For instance, tryptophan is shown feeding into both pyruvate and acetyl-CoA. And that's because different parts of the molecule, after they're broken down, feed into different intermediates. So part of the tryptophan molecule goes into pyruvate, part of it ultimately goes into acetyl-CoA. And this is a table showing the reverse of that for the non-essential amino acids and what intermediates can be used to synthesize those if it's required. Okay, again, we want to remember that some amino acids feed into sugar precursors, and those are the glucogenic amino acids that can generate glucose, and those are listed on this slide. There are also ketogenic amino acids that feed into the TCA cycle. Then there's a list of those that are considered both glucogenic and ketogenic amino acids, and that's for the reason I mentioned a minute ago, that part of the molecule, when it's broken down, can feed into pyruvate, and the other part of the molecule goes into some other intermediary in the TCA cycle. So this is a really busy slide showing that. If you're interested, I don't think there's any point in memorizing all this. Again, I think it's just good to know that some amino acids can be used to maintain the level of glucose available, where others can't, even though they can be used to generate energy. They can't be used to synthesize new sugar, new glucose. And this shows a schematic of precursors, again, that can be used to generate the non-essential amino acids. And notice here that arginine is on the list here. So in this figure, arginine is considered a non-essential amino acid. So tryptophan and phenylalanine are the most complicated amino acids, so you might imagine they are the most complicated to break down. So this shows the pathway for both of those. Again, feeding into ultimately into the TCA cycle, and tryptophan though can break down into alanine, which can feed into pyruvate. Now I'm showing you this because I want to talk a little bit about inherent defects in amino acid metabolism, and there are some in the breakdown of these amino acids, but there are others as well. And one of them shown here is what's sometimes called maple syrup urine disease, and it's called that because what happens is people with this problem cannot break down the non-polar amino acids, and they tend to have a sort of a sweet smell or even a sweet taste. So they're excreted in fairly high concentration in the urine, giving it that sort of sweet smell. Also, babies who have this problem, I think if you kiss them or touch their skin, it's actually sweet from sweat glands excreting the amino acids. Okay, so if these go untreated, they can lead to some pretty serious problems, both physical and mental retardation. And the best way to deal with it is to very close control of the diet to limit the number of these amino acids. I'm not sure the supplementation with high doses of thiamine, how effective that is, but it is used. Okay, so the aromatic amino acids are involved in production of a number of different molecules, some of them shown here, and there are some defects in just about all these pathways that lead to different uh, uh, issues. The first step in phenylalanine, well, in this case, phenylalanine conversion to tyrosine, if we can't do that, it can cause a number of issues in terms of pigmentation of the skin and as well as production of dopa or other catecholamines. Now, if you have difficulty in breaking down phenylalanine and metabolizing it, what's going to happen is you're going to make an excess of tyrosine, and that's going to be used to make a excess of other molecules that it serves as a substrate for. For instance, melanin or pigment, skin pigment, and this can result in some serious medical issues that are listed here. Okay, so tyrosine is also a precursor for melanin, and if you have a defect in the enzyme tyrosinase that produces melanin, then you'll affect albinism, which of course is lack of pigmentation. Generally, this doesn't involve a lot of other serious medical effects. Now, phenylalanine can be converted into not just tyrosine, but dopa and dopamine, which are neurotransmitters. There are naturally occurring substances that mimic those neurotransmitters. One shown down in the right-hand corner. Mescaline is a agonist of dopamine receptors and can cause some interesting side effects. The same is true of tryptophan, which can be converted into serotonin, another neurotransmitter, and there's a semi-naturally occurring compound, LSD, that is an agonist of serotonin. Interestingly, it's also interacts with dopa receptors, so it's very complicated. And there are a number of serotonin receptors, and LSD binds to some of those and not others. Psilocybin is another naturally occurring compound that is also a tryptophan analog, which interacts with serotonin receptors. So I want to come back to this idea once again. If we have excess of amino acids, or if we are looking for a source of energy through dietary protein, those can be, amino acids can be deaminated. The skeleton, then carbon skeletons, can feed into either the TCA cycle directly or through pyruvate, which then can go in either direction through the TCA cycle or back into glucose. And there's then this direct connection between the urea cycle, which is used to get rid of the excess nitrogen, and the citric acid cycle, or TCA cycle, through the arginosuccinate. I want to talk about one more group of nitrogen-containing compounds, and these are derived through from this compound, ornithine, which is part of the urea cycle, but it can also generate a group of compounds that we call polyamines, and those are shown here. Again, at the top is the precursor for polyamines, ornithine. If we remove the carboxyl group, Um, on the ornithine, we have putrescine. Cadaverine is derived from a different pathway because there's an extra carbon in the chain, but spermidine and spermine are then derived from putrescine by adding an additional 3-carbon chain with another amino group onto one side for spermidine, and then adding an extra one on the other end for spermine. So this is the pathway, starting again with ornithine. The enzyme ornithine decarboxylase removes the carboxyl group from the ornithine to produce putrescine, and this has been a target for cancer therapy because if you inhibit ornithine decarboxylate or ODC, it tends to inhibit cell growth because these compounds are required for cellular growth. And so ornithine decarboxylase is the rate limiting step. It's been a target for therapy. Once we get to putrescine, then again, the 3-carbon chain with an extra amino group is added using this S-adenosylmethionine to produce spermidine, and then if we add the same 3-carbon amine to the other end, we get spermine. So again, ODC or ornithine decarboxylase is upregulated in a wide variety of cancers, and not just in cancers, but any fast-growing tissue is producing a lot of polyamines, and so they're upregulating ornithine decarboxylase. And in fact, when I was an undergraduate just a few years ago, I worked in a lab that studied polyamines, and we were always looking at polyamine synthesis in a wide variety of fast-growing tissues, even in certain plants, like if you're familiar with the agave that some of you might be familiar with, its distilled product called tequila, but it's also called a century plant, not because it lives for a century, but it lives about 30 or 40 years, and then it sends up this flowering shoot out of the middle that can be as tall as 15 or 20 feet, and that grows very rapidly. Some people even claim they can see it growing, although I'm not sure that's true. But we were actually measuring polyamine synthesis in the growing stalk. Once that stalk matures and flowers, sends out its seed, then the plant dies. Anyway, back to polyamines. ODC is the rate limiting step in synthesis, and there are a number of drugs that have been used to target the activity of ornithine decarboxylase in treating cancer. And I think there's been a variety of success. I think it's been successful in some cases, but not as a broad spectrum use of, or broad spectrum treatment of many different tumor types. Okay, so that's what we need to talk about for amino acid metabolism. We're going to move on next time and look at some, go back and look at some complex carbohydrates.